Good morning everyone. So welcome to my YouTube channel. So in CSIR UGC license syllabus, so there is a module called systemic physiology, plant physiology. So I am going to explain the concepts present in the plant physiology. You have photosynthesis. It's very important. Every year they are asking C part, B part question from this area, and respiration and photorespiration, nitrogen metabolism, plant hormone, sensory biology, solute transport and photoassimilation translocation, that is nitrogen metabolism and transport, secondary metabolites, stress physiology. So these are the modules, the units you can see in the module systemic plant physiology. And this plant physiology is a very high scoring module in CSIR question paper. You will get at least 5 to 6 questions from CPAL, and you will get 4 to 5 questions in B part also, because uh, plant, if I take as a holistic way, in the development biology also there is a topics in plants, that is a double fertilization, zygote formation, and the symmetry of plants, seed formation, different hormones in seed formation, then phyllotaxy, Arabidopsis, ABCD model, ABCE model, then transition from flower symmetry, the flowering mechanisms, then embryogenesis. Embryology is a very important topic. I think in last year there were questions from this area also, embryogenesis, various genes, shoot and root development, apical development, transition to flowering, floral meristems, so everything. So when we consider plants, both from sixth module and development biology, together you will get lot of questions from this. I think some 20-25 marks you will definitely get from the plant area. So today we are going to study about sixth module, plant physiology, and the subunit is sensory biology. This is the topic that we are going to discuss. Here you will study about phytochromes, cryptochromes, phototropins, and stomatal opening, photoperiodism, biological clocks, etc. So C-part questions, many C-part questions are there from this type. So we will see that. So these are some of the questions that I have taken randomly from previous question papers. See COP1, cryptograms. This is a C-part question. Phytochrome. This is another year. Phytochrome mediate control of photomorphogenesis. COP1, E3 ubiquitinase. Hi-Fi. What is Hi-Fi? So this is another question. This is a first computer-based CSI, I think 2019 June. They asked regarding the phytochromes and chlorophyll. This is another C-part question. Uh, Photoreceptors, Arabidopsis thaliana. See, this is also the same question. Okay, this question was asked in another year, and this is another year. So in CSIR question paper, what I analyze is that there are many repeated questions. In addition to that, many questions are reframed. So this is another example of reframing of questions. See, then red light, far light, phytochromes, another C-part question. See, this is light perceived by various photoreceptors, phytochrome A, B, cryptochromes. See, these all are C-part questions I have randomly chosen. So this is a very thing. So after learning or after watching my video, you will be learning about this photoreceptors. Okay. So I'm following the Taiz-Zeiger 6th edition. I think this text is the best textbook for plant physiology. I think if you learn this Taiz textbook completely, I think you will be able to score full marks in plant area in CSI biology, because most of the questions they are directly, some they ask directly, they are asking from this textbook. So you have to study this Taiz-Zeiger full text, full first page to last. Don't worry, I will help you in learning the subject. Now, so you all know that you all know about photosynthesis, isn't it? So what is photosynthesis? Yeah, photosynthesis is actually uh, the preparation of food by plants with the help of sunlight using carbon dioxide. So this is a simple definition you are learning from the lower class itself. So there in the photosynthesis, actually sunlight acts as a energy source for photosynthesis, isn't it? As a light energy for water splitting, for electron excitation from uh, one what we can say complex to another. So they act as energy source. But other than this energy source, sunlight also acts as a signal for various developmental processes in plants. That is from seed development in senescence, various light mediated response. So all these things, sunlight acts as a signal. Okay. So to capture this signal, that is to capture the sunlight, plant needs something, isn't it? So that thing is the photoreceptors. So what is photoreceptors? Photoreceptors, they are the receptor molecules present in plants for what? To detect sunlight. That is the photoreceptors. So what this photoreceptor do is that they will absorb photon of given wavelength and use this energy as a signal that initiate various photon or light mediated responses or photoresponses we can say. So this various signaling include light regulated growth, okay, uh, then chloroplast movement, stomatal movement, phototropic movement. So everything the sunlight will act as a signal. Okay, and this signal is captured by the receptor called photoreceptors. So that is the thing that photoreceptors that we are going to discuss in this lecture series, plant photoreceptors. So you can see photoreceptors, they will absorb photon of given wavelength and use this energy as a signal to initiate various photoresponses. So when we consider a photoreceptor, actually, photoreceptor consists of a protein part and a non-protein light absorbing part. So the light absorbing part of photoreceptor is the non-protein part. So that we call it as chromophore. Okay. So all the photoreceptors consist of a protein part and a non-protein part. Actually, except UVRH. UVRH do not contain this protein and non-protein part. Okay. So what does photoreceptors do? Photoreceptors can sense light intensity. They can sense light quantity and duration and exposure of light. See, in the textbook you can see they can sense light quality, light quantity, light intensity, duration of the exposure of light. So for all these things, uh, they can sense these things and sunlight acts as a cellular signal to regulate various specific photoresponses. Now, next is five types of photoreceptors. So what are the types of photoreceptors? How many types are there? Okay, so you know that the photoreceptors, there are different types of lights, isn't it? Different wavelength of light. There are red light, there are blue lights, there are green light, UVB light. So these lights have different wavelengths. Okay, so these photoreceptors uh, are also classified to different types based on their ability to absorb specific wavelength of light. So based on their ability to absorb specific wavelength of light, photoreceptors are classified into five major types. First one is phytochromes, second is phototropins, third is cryptochromes, pseudo, and UVR8. First one is phytochromes. Phytochromes actually they will absorb red and far red light. They can also absorb blue and UVR light. Okay. Then cryptochromes, phototropins and seed loop, they will absorb blue light. And UVR8, as the name indicates, they will absorb UVB light. So UVR8 receptor, they will absorb UVB light and they will induce UVB induced photomorphogenic response like flavonoid synthesis. It's a secondary metabolite. It is mediated with the help of UVB light. So these receptors, UVR8 receptor, help in mediating this kind of responses. Okay, and cryptochromes, cryptochromes, they are important for flowering, especially cryptochromes 2. They promote flowering. And the phototropins, they are involved in phototropics responses, phototropism. Then chloroplast movement. It's very important. It is given in your CSIR syllabus also. Questions are coming from CPAR questions. Then stomatal opening. How stomato open? They all depends on this uh, phototropins, blue light phototropins receptors. Now, actually, the sunlight they will act as a sunlight will act as a signal, and the signal is received by the photoreceptors, isn't it? And there will be some signal mechanisms, and the plant will undergo certain morphological changes based on the signal or based on the photoreceptors or based on the wavelength of light, what type of light, whether it is blue light one response or they will mediate certain morphological responses. Okay, so the light mediated morphological responses or we can see this morphological responses can be of two types. First one is photomorphogenic response, photomorphogenic response or the photomorphogenesis. Second is a scotomorphogenesis. Photomorphogenesis. So as the name indicates, photo, light. Okay. So we have light and this light is sensed by who? Photoreceptors, isn't it? Then we have the signal, we have the receptor, then there will be signal transduction, then they execute certain morphological changes. What are the changes? First one is short hypocotyle. Second is open cotyledon. So short or hypocotyle means, see if there is the presence of sunlight, the stem or the hypocotyle will be short because these plants do not want to go outside and search the sunlight. So they need only short hypocotyle. Okay. And they have open cotyledon. What is cotyledon? It is a first emerged leaf, we can say. So for because we have light, the plant can synthesize photosynthesis. So they have open cotyledon and short hypocotyle because they don't want to search in light. So they don't want an elongated hypocotyle. But in case of scotomorphogenesis, that is in the absence of light, the plant will undergo certain morphological changes. That is, they have elongated hypocotyle, or it is called etiolated. Elongated hypocotyle because it is the absence of light. So they want to search for the sunlight, isn't it? So see if you keep, for example, if I say, if we keep seeds in the dark, so you will see that after the germination, the hypocotyle will be elongated because they are in search of light. So they have elongated or etiolated hypocotyle, and their leaves are also not photosynthetic, not green. You will see some light white colored cotyledons. See, this is the example I'm selling. So this is the plant that they have kept in light. So light, it is sensed by the photoreceptors. So they have short hypocotyle. isn't it? Because they have light, they don't want to uh, go search in capturing light. But this is a plant they have kept in the dark. So you can see it's a elongated hypocotyl because they are in a search of capturing light. Okay, and you can see the cotyledon. The leaves are also non-photosynthetic, not green. Here you can see phototropic cotyledons, isn't it? So that is about the morphological responses that they play. Now, sometimes this photoreceptors, that is the phytochromes and cryptochromes, together they will act to mediate certain responses. Phytochromes and cryptochromes, they are two different photoreceptors. They together with core action, they will mediate or they will execute certain photomorphogenic responses. For example, if you're if you keep a dark plant, if you keep a dark if you keep a plant in dark for a long time, then if you expose to sunlight, there will be de-etiolation. So the chlorophyll will respond and green colors will regenerate. So this is an example of co-action of this two photoreceptors, phytochromes and cryptochromes. Okay. Now we are going to see the different wavelength. So the types of radiation. So this is a visible light. You can see 715. This is a red light. Okay. So blue light, green light. This is UVRB 8. So based on the light, we have different photoreceptors. Red light far field absorbed by photo phytochromes. Blue light is absorbed by cryptochromes, phototropins, and UVB light is absorbed by UVRH. Now, see, this is an experiment. I think this is asked from last few years back. One CPAL question was asked here. So here you can see the seeds are placed in dark. So there is no seed germination. Okay. Now when seeds are exposed, the seeds are given red light, there is seed germination. Seeds able to germinate in the presence of red light. Then the next experiment, the red light followed by a far red, the seeds do not germinate. Okay. Then next experiment, red light followed by far light, again red light, seed germinate. Next experiment, red light followed by far red, then red light, then far light, seed do not germinate. What does it mean? It means that far red light inhibits seed germination and red light promotes seed germination. Okay, so you can see that here red light promotes seed germination, and the last light, you have to see the last light to detect whether seed germinate or not, because in some part the C part question they ask the seeds are treated with red light, far red light, then what is the response, like that someone question was there. So here red light followed by far red, seed do not germinate. So far red actually inhibits seed germination, then far red, then again red light is given, seed germinate. So you have to see the last light, which is the last light, then only you can see, you can predict whether the seed will germinate or not. So concludingly we can say that far red light will inhibit seed germination, red light promotes seed germination. So this is a light mediated signal, isn't it? Light mediated signal, other than photosynthesis, this is an example of light mediated signal. Now, actually, how they found that red light will promote seed germination or far red light will inhibit seed germination. So in this graph, you can see relative quantum effectiveness with wavelength. They got that in red light, they got a stimulation hike. But in far red light, they got an inhibition hike. From this, they conclude that red light promotes seed germination and far red light inhibits seed germination. Okay, now we can see more about phytochromes. So as I said that based on the wavelength of light absorbed by the photoreceptors, photoreceptors are classified into various types. First, we are going to discuss about the phytochromes. So actually, phytochromes are they are the red or far red light absorbing photoreceptors. They execute various photomorphogenic responses. Phytochromes are actually cyan blue or cyan green protein with molecular mass about 125 kilodalton. Phytochromes actually exist in two forms, in PR form and PFR form. PR form is a red light absorbing form and PFR is a far red light absorbing form. So in PR, they will absorb red light and it is converted to the PFR. So PFR is the active form. Okay. When PFR absorbs far red light, it is converted to the inactive PR form. So this is very important. Based on this concept, some questions are asked. Okay. So this is called as a photoreversibility. That is, when PR is absorbed by red light, it is converted to PFR, and when PFR absorbs far red light, it is converted to the inactive PR form. Okay. So when PR, that is the inactive form, absorb red light, it is converted to PR form. So actually 12% of PR exists in the inactive form, while the conversion of PFR to the active form. So only 88% only the active PFR is maintained. The transition to PFR to PR, actually they absorb forward and PFR is converted to PR. So actually 2% remain in PFR form while converting to PFR to PR forms. So 98% it is converted to PR form while absorbing the forward. So this process actually it is called as a photostationary state. That is an equilibrium is maintained. Okay. So PFR, as I said earlier, it is a physiologically active form of phytochrome. So as I said that this phytochrome, like all photoreceptors except UVA rate, they contain a protein part as well as a light absorbing chromophore, isn't it? So when we consider the chromophore, the phytochrome chromophore is called phytochromobilin. It is represented by P5B. Now the protein part, it contains an N-terminal domain as well as C-terminal domain. Sorry, this an N-terminal domain and C-terminal domain. Now coming to the non-protein part, that is a phytochromobilin. It is a linear tetrapyrrole ring designated as ABCD. Okay. So actually this phytochromobilin they are synthesized in the plastids. You know the plastid chloroplast, it's present in the cell and is derived from heme. Okay. So you know that there is a protein part as well as a non-protein part. So this non-protein phytochromobilin is attached to the protein part to cysteine residues present in the protein part via thioether linkage. That is, there should be an attachment between protein and non-protein bond, isn't it? So here the phytochromobilin is attached to the cysteine residues present in the protein bond through which linkage? Thioether linkage. Now so I will show you the textbook. Actually, this is the phytochromobilin structure. We will discuss more about that, don't worry. Sorry, this is actually the phytochromobilin. This is the non-protein bond. So actually it is synthesized in the plastid. As I said earlier, it is derived from heme. Heme is converted to biliverdin in the presence of hyphae gene, and biliverdin is converted to phytochromobilin in the presence of Hi2. Okay. After the formation of phytochromobilin, it is transported to the cytosol. At the same time, in the nucleus, the protein part also is get transcribed to mRNA, and it comes to the cytosol, and the translation occurs, and the protein part, that is phytochrome apoprotein, will combine with the phytochromobilin and they form the active phytochrome. Now coming to the protein bond, it contains an N-terminal domain as well as a C-terminal domain. So N-terminal domain, it contains various domain, pass domain, graph domain and phi domain. Okay. So there is a code to remember. Pasai Gafur Physicsil. So Gafur is the name of a guy and he has pass in the subject physics. Pasai Gafur Physicsil. It's a Malayalam. I'm a Malayalam guy, so I'm using this code to remember the N-terminal domain of protein part of phytochrome, pass domain, graph domain and phi domain. So pass domain, actually they don't have much role, or it's not yet discovered, and graph domain contain a bilin lyase domain. So through this graph domain, actually phytochrome binds. You know that phytochromobilin bind. You know that phytochromobilin is a non-protein bond, isn't it? So it has to be attached to the protein bond. This is the protein bond. So in the N-terminal there is a graph domain. Through this bilin lyase activity, the phytochromobilin get attached. Through which linkage? Yeah, thioether linkage. Then phi domain actually stabilize phytochrome in the active PFR form. Then coming to the C-terminal domain, it has two PRD domain, that is pass related domain, and one HKRD domain. Actually PRD domain serves as a NLS signal. What is NLS signal? It is nuclear localization signal helping mediating dimerization. Okay, so because why they want actually this NLS signal? What is the purpose of this NLS signal? Actually, as I said that this photoreceptors or phytochromes, they will absorb sunlight as a signal and they will mediate certain response, isn't it? So for mediating certain response, they have to transcribe certain genes. So this is the protein part, we can say. See, this is the protein part, this is the N-terminal domain and this is the C-terminal domain. Okay, so N-terminal domain contains three domains. Sorry, N-terminal part contains three domains: pass domain, graph domain, phi domain. And graph domain contains bilin lyase activity, through which this phytochromobilin, the non-protein part, is getting attached. Okay, and the C-terminal domain contains two PRD, pass related domain, and one HKRD domain. Okay, actually I said earlier also, the phytochrome, actually they are active in PFR form. In PR form they are inactive. So this is an inactive form. So when they get red light or when they absorb red light, they are converted into PFR form. Okay, so actually what is happening is that during the PR inactive form, actually here they have a NLS signal in the PRD domain. So this PRD domain is getting masked. Okay, so when they absorb red light, this PRD domain is exposed. So there is a conformation change here. So they will move into the nucleus and they will transcribe, and when they will certain genes. What type of genes? Photomorphogenesis response. So there is a signal sunlight. It is absorbed by photoreceptors here, phytochromes. Okay, phytochromes. Then they will absorb red light, and it is converted to PFR, the active form, and is enter into the nucleus. Then gene expression, then photomorphogenesis response. It can be like chloroplast movement, or here phototropism. So it can be for phototropics like bending, various phytochrome related response. It can be any morphological changes like shoot elongation or whatever it may be. Now, here you can see the gene expression, and it execute various photomorphogenesis. So what are these phytochrome responses? That is rapid biochemical events, slower morphological changes including the movements, growth, etc. Now we are going to see, as I said earlier, PFR is the active form of phytochrome and PR is the inactive form. When PR absorb red light, they are converted to PFR, which is the active form of phytochrome, isn't it? Actually, what is the conformation change that is present in PR and PFR form? So, actually, the phytochromobili, this we are discussing about the phytochromobili. So the non-protein part of the phytochromobili will undergo certain conformational changes when during the conversion of PR to PFR using red light. So what is the conformation change is that initially it exists as 15-cis linear tetrapyrrole. 15-cis linear tetrapyrrole. Oh, sorry, it is the picture. Initially there exists as a cysteine-cis linear tetrapyrrole. So when they absorb red light, so this C14 and C15 bonds, sorry, C14 and C15 bonds rotate. That is, rotation of D ring occur, and while rotation of this D ring, there occurs certain conformation change and is converted to the active PFR form. Okay, so this is actually one model they are explaining what are the conformation changes. This is one model, and second model is actually the Tugel model. We call it as a Tugel model. This is a Tugel model. So in Tugel model, they are saying that when PR absorbs red light, it is converted to PFR form. Okay, so the PR actually exists in a beta hairpin ring. Okay, so when they absorb red light, it is converted to alpha helix, that is a PFR. Then this PFR, this is the active form. When they absorb far red light, they are converted to beta hairpin. Okay, so these are the conformation change in protein occur during this conformation. So this is two models they are explaining the test. We don't know which is the accepted model. They have these two are maybe possibly it is a proposed model we can say. Okay, now we are going to discuss the various phytochrome responses, some phytochrome responses. So phytochrome responses can be of three types: very low fluorescent response, low fluorescent response, then high irradiance response. Actually, very low fluorescent response, it is a response initiated by fluorescence as low as 0.0001 new mole per meter. That means very low light. Okay. So example is Arabidopsis seed germination is an example. Here, no photoreversible. It is non-photoreversible. That means I told you that when PR is converted to PFR, actually the PR, while converting PR to PFR, actually 12% remain in the inactive form. Only 88% is converted to PFR, the active form, while absorbing the red light. Okay, so here the light is 0.0001 mole, so they can't remain like some 12%. No, it's not possible. So here not photon reversible. Then here VLFR obey law of reciprocity. Now what is law of reciprocity is that, see, the bright light mediates certain response, isn't it? So the long exposure of bright light executes certain response. It is same as the dim light exposing for a long time. Once more I will explain. The bright light exposing for a few minutes or few time execute certain response. So this response will be similar to the dim light exposing for a longer time. So this is called law of reciprocity. So in case of VLFR, law of reciprocity is applicable. Next is low fluorescent response. It is a response, the fluorescence is 1 new mole meter square. Example is lattice seed germination, inhibition of hypocotyl elongation. These are the examples. Here law of reciprocity is applicable, and here photo response is reversible because it is 1. So they can convert to 12 to 80% is possible. Next is high irradiance response. So it is high irradiance means the response initiated by high irradiance, that is 100 times more irradiance than LFR and FR. So examples are the flavonoid synthesis, secondary metabolite anthocyanin. So they need high irradiance. So these are some plant photomorphogen response induced by high irradiance. See, synthesis of flavonoid, inhibition of hypoelongation, induction of flowering, then enlargement of cotyledon and mustard. Actually, before coming to this VLFR, LFR and HIR, actually I should familiarize you with two terms which I have forgot. Actually, what is this fluorescence and what is this irradiance? Listen, you will be confused. What is this fluorescence and what is this irradiance? So, fluorescence means it is the amount of light imprinting in a unit surface area. The amount of light imprinting in a unit surface area. In a unit surface area, how much the light amount is imprinted. That is the fluorescence. Irradiance means the amount of light received per unit area per unit time. That means here in irradiance time is also considered. Okay, so in per unit area per unit time, how much amount of light is perceived, that is the irradiance. So based on fluorescence and irradiance only, the phytochrome mediated responses are classified into three types, that is the VLFR, LFR and high irradiance. Very low fluorescent response, that is 0.001 new mole. That is example is Arabidopsis seed germination. Second is low fluorescent response. Fluorescence is 1 new mole meter square. let us see germination. Next is high radiance response, that is 100 times more than LFR and FR. The synthesis of flavonoids, certain anthocyanin, etc. Here they obey the law of reciprocity. Okay, so this phytochrome actually exists in 5 isoforms. Phytochrome A, phytochrome B, phytochrome C, phytochrome D and phytochrome E. So phytochrome A is actually unstable. They control plant responses to LFRs. Okay, and phytochrome B, C, D, E, they are light stable phytochromes. Actually, phytochrome D and phytochrome E, they mediate petiole and internodal elongation. That is a phytochrome mediated response mediated by phytochrome D and E. And phytochrome A and phytochrome B, they don't have a nuclear localization signals. Okay, they don't have a canonical nuclear localization signal. So in case of phytochrome B, you know that they have PRD domain, isn't it? Pass related domain. So this PRD serves as a nuclear localization signal. Okay, so that means this phytochrome B, actually they are PR form, inactive form. That is the nuclear localization signal is masked. When they get light, they are converted to the active PFR form. So this nuclear localization signal is exposed. So they facilitate phytochrome B to nucleus. Then the nucleus, there will be certain gene expression and photomorphogenic responses will be take place. But phytochrome A, they do not contain this PRD or they don't have this nuclear localization signals. So two proteins helps in this transport, that is FHY1 and FHL1. So these two will help in the transport of this phytochrome A to the nucleus, and then they can mediate the photomorphogenic response. Now, I told you that this phytochrome regulates gene expression. Actually, how this phytochrome regulates gene expression? That is the thing that we are going to discuss now. You have to study about PIF. PIF is actually phytochrome interacting factor. Phytochrome interacting factor. I will show you the text. Phytochrome interacting factor, PIF. Actually, what this PIF will do is that PIF is actually a negative regulator of photomorphogenic response, as well as it is a positive regulator of scotomorphogenic response. What is scotomorphogenic response? It is a dark mediator response. So PIF actually acts as a negative regulator. You can see PIF acts as a negative or inhibitor of photomorphogenic response, and PIF will promote scotomorphogenic response. How? I will show you the textual image. See, so the first case, it is regarding the scotomorphogenic genes. Okay, so you know that phytochromes in dark, this is a moon, that means it is a dark. So phytochrome in PR form, they are inactive. Okay, so PIF will so PR is inactive, so PIF is free. PIF is free, so they will interact with the scotomorphogenic genes, and they will transcribe these genes, and it will result in the scotomorphogenic response. Okay, but in the case of light, you know, here it is the sunlight is given. So in the presence of light, the light will convert PR to PFR. You know that PFR is the active form of phytochromes, isn't it? So this PFR will degrade PIF. See, the PIFs get degraded. So the PIF cannot interact with the scotomorphogenic genes. So this transcription of scotomorphogenic genes is inhibited, Hence, no scotomorphogenic response in the presence of light, isn't it? See, in the presence of light, they should undergo photomorphogenic response. So there is no need for scotomorphogenic response. Now, coming to B diagram, actually in dark, so here they are showing regarding the photomorphogenic genes, photomorphogenics. In the first image, they have given scotomorphogenic genes. Here it is a photomorphogenic genes. So in the dark, you know that phytochrome exists in the inactive form, that is a PR form. Okay. So the PIF is free. So PIF will bind to the photomorphogenic genes and sorry, they will bind to the photomorphogenic genes, and this PIF will act as a repressor for photomorphogenic genes because it is dark side. It is a dark, so they don't want to mediate any light mediated response or photomorphogenic response. But in case of or in the presence of light, this you know that PR will be converted to PFR active form in the presence of light. So this PFR will degrade PIF. So the PIF cannot act as the transcription repressor of photomorphogenic genes. So this photomorphogenic genes will be transcribed and photomorphogenic responses will be mediated in the presence of light. Okay. So that is how this phytochrome will mediate uh, photomorphogenesis. Now, I told you that this phytochrome induce various photomorphogenesis responses. So the phytochrome induced photomorphogenesis involves the degradation of certain proteins. Degradation of certain proteins. So which are these genes? See, phytochrome-induced photomorphogenesis involves protein degradation. Okay. So before coming to that, so COP, DET, FUS, these are the certain genes. These genes will include COP1 and SPA complex. And these genes will also encode CNS complex. See, it is very important. I think a few years back a C-part question was asked from this area. Okay, so COP1 and SPA1 together they will add ubiquitin tag to photomorphogenic response genes. So which are photomorphogenic responses here? Hair, laugh, high. These are the photomorphogenic response genes. Now coming to the textbook. See, this is the dark side and this is the light side. This is the night and this is the morning time. So what happened in the night? So in the night actually carbon exists in the cytoplasm. So this is actually the cytosol. This is the nucleus. First you have to understand. That is the first thing you have to understand. And in the top portion they have shown about the sunlight, that is the morning, and in the bottom portion they have given the moon, that is during the dark time. So during the dark time actually carbon first initially exists in the cytosol. This carbon is transported into the nucleus with the help of CSN. CSN complex will transport COP1 from cytosol to the nucleus in the dark. So in the nucleus, this COP1 will bind to the SPA1 and they will add ubiquitin tags to the HFR, LAF1 and Hi5. Actually HFR, LAF1 and Hi5 they are the photomorphogenic regulating genes. Okay. So in the dark actually these genes do not want to express because it is the dark. These genes are needed only in the presence of light. So they will add ubiquitin tag and it is degraded by 20S proteasome and this is degraded lit. You know this degradation process. Okay. Then in the morning or in the presence of light, actually carbon, actually they are present inside the nucleus, isn't it? In the night they are present in the nucleus. So this carbon will move out to the cytosol from the nucleus. So since carbon is not or carbon is absent, so there is no one to add ubiquitin tag to HFR, hyphae and LAF. So what are HFR, hyphae and LAF? I told that they are the photomorphogenic regulating genes. Okay. So since no one is there to add ubiquitin tag, these genes are free and uh, they will act as a transcription regulators and they will bind and transcription occur and gene expression also. The photomorphogenic responses genes will be expressed and photoresponse also will be mediated. That is in the presence of light. Okay, so this is very important. I think, as I said earlier, C-part questions you will expect based on this concept. So that's all about phytochromes. So we have studied about phytochromes, the phytochromes, their responses, isn't it? Their PRPFR form conversions. Then we seen about their protein part, non-protein part, the phytochromobilin. Then there are conformation change that occur when PFR is converted, PR is converted to PFR form. Then what are the domains in protein parts? Then we saw about the PIF mediated responses, cotomorphogenesis and photomorphogenesis. And also we saw about various phytochrome responses, VLFR, VLFR, HIR. Then we saw how phytochrome regulates gene expression. Then what are the mechanisms, that is the protein, how the protein degradations occur when photomorphogenesis responses occur. So these all things we have seen about the phytochromes. Next topic is the blue light response or cryptochromes, or the blue light responses and photoreceptors. That is, which are the photoreceptors that absorb blue light and they, how do they mediate this blue light mediated responses like chloroplast movements, stomata. It's very important for CSI questions. So that we will discuss in another video. So if you like this video, so please like my video and share my video to get the next part of the video. So within 1 week we will finish off all this plant physiology. It's a very simple Thais textbook and you will get good marks in the plant physiology topics of CSIR. Thank you so much. So please like and share my video.